My name is Marcus Mahl. I'm the chair of the Pediatric Pulmonology and Immunology Critical Care Medicine and the Cystic Fibrosis Center here at the Charité, and have a long-standing interest in cystic fibrosis, also development of personalized medicine with project at the BIH, and it's therefore a great pleasure to welcome our speaker of the BIH Multi-Omics Lecture today, Dr. Jeff Beekman from the University Medical Center in Utrecht, who will talk about primary epithelial cell models for cystic fibrosis studies, which both is highly relevant and interesting for the cystic fibrosis and rare disease community, as well as the development and use of patient-derived primary cell models for translational research. Before we start and I introduce Jeff, I want to make some housekeeping notes and remind you again that you please turn off your webcam and microphones at any time. We want to have a lively discussion at the end. Please ask your questions through the chat function you can find at the right upper panel of the website, and send these to the organizer, and then I will get them. And also please note that the lecture will be recorded for the BIH. With this, I'd briefly like to introduce Jeff. He's a full professor of cell disease models at the Department of Pediatric Pulmonology at the University Medical Center Utrecht in the Netherlands, where he has established a translational research group that focuses on the development and validation of patient-derived cell models for basic studies, drug development, and individualized medicine applications. He's been trained in molecular and cellular biology in the context of immunology initially, then became independent in 2010 when he started focusing on cystic fibrosis, and really pioneered the development and use of 3D stem cell models, primarily intestinal organoids for functional assays, where he published a seminal Nature Medicine paper in 2013, where he showed that organoids can be used to study CFTR function and response of emerging therapies that target mutant CFTR. And since then, I think he's published more than 50 papers in the last 10 years on this topic. So we've been really a leader of this field, which is also shown by several awards he received from the European Cystic Fibrosis Society, European Respiratory Society. And at the moment, in addition to his work on CF, he also aims to expand his disease models to other monogenetic diseases, as well as make use of them in the context of to better understand infection within the coronavirus. So without further ado, Jeff, I want to hand over to you and look forward to your talk.
So I think we should now be able to see my presentation. It's a big pleasure to be here. Thanks, Marcus, for the nice introduction. I'll just kick away and start because I have a decent slide deck. So I'll try to stick within 45 minutes, so we have some time for discussion as well at the end. I will mostly be talking about intestinal organoids and how we use them actually for modeling of cystic fibrosis, for drug development studies, but mostly also to see how well they can be used for personalized medicine settings. At the end of the talk, I also will mention some of our work with airway organoid models, but it will be very briefly, but we can discuss if this is needed. Well, let's then continue. So this slide just illustrates how we work in science, obviously. So we do clinical studies, we calculate averages, but in real life an average obviously does not exist. We're all individuals, and this just shows this nicely illustrates this concept. This is, you can see the average phase of all these individual phases. And obviously, if we develop compounds or drugs for therapeutic use, efficacy always based on averages, while the individual efficacy can be pretty, well, because we're all different, individual efficacy can be rather different. So already some 10 years ago, we tried to develop new ways to better understand and predict the individual disease and therapeutic response of people. And we considered that it would be nice to use laboratory culture models to do this, because this allows you to get an individual patient sample into your lab as a living entity and stimulate it and use it for various research purposes. So at that time, it was not so easy actually to do cultures. At that time, iPSCs were being developed at, well, I think at well, massive scale now, but that was really hot at that moment then. And another culture model that was being developed were intestinal organoids then in the lab of Hans Klevers. He is our neighbor, or he was located in a neighboring institute. And these are different because if you compare these to iPSC, then these are reprogrammed cells, which are really pluripotent, while these are adult stem cell based cultures in which you really retain the tissue specific epigenetic profile. So whereas you can generate many tissues from these stem cell sources, this is a three-dimensional tissue in which you have adult stem cells which can differentiate to different types of tissue resident cells, but it remains committed to the intestinal compartment. So we use this to study cystic fibrosis. I think many people here know that cystic fibrosis is a disease of an ion channel. I think it's now, I lack the cartoon obviously now, but it's an ion channel which leads to chloride secretion in many epithelial organs. Most clearly, the lungs are affected, but also along the gastrointestinal tract. Actually, you see that the epithelial secretions are being altered, and that you mostly also in the lungs, but also in other tubular structures, you get thick mucus and accumulation of thick mucus, which leads to stasis and clogging up the epithelial secretions. And especially in the lungs, you see that there is a bacterial accumulation and large high-grade inflammation, which leads to tissue destruction. So the mucus is really thick, as you can see here, and most people with CF eventually succumb to end-stage lung disease. But but definitely the disease is far more broader. It's systemic. Lots of epithelial cell systems being affected, but also, for instance, the sweat glands and the reproductive tract. What's really, I think, really exciting in this field over the last 10 years also is that we now see the development of modulators which really target the genetic defect in CF. So CF is monogenic disease in which you have an ion channel that does not function, and there are now lots of small molecules which can really restore the function of this ion channel. So they can bind to the faulty protein and reintroduce a conformation which is functional again, and then by this restore function of CFTR. So what you see here, this is pretty a complex cartoon now. What you see is that normally CFTR would be present here at the cell surface, where it leads to secretion of ions such as chloride. But there are really a lot of mutations in CFTR. There are over 2,000 variants known. They're estimated that about 1,700 of these variants are CF causing. And although this is a really large number, the majority of alleles is mostly F5ODEL. So this is a single type of mutation which in which a phenylalanine lacks at position 508 in the protein, which leads to mistrafficking and accumulation in the ER upon synthesis, misfolding, and degradation through the lysosomal or proteasomal pathway. So this is a mutant which hardly reaches the cell surface, There are some other 10 other mutations which have a pretty high prevalence, but the majority of these 1700 mutations are really rare and unique, and they partly, but they're all only present in a smaller fraction of CF patients. If we think about the new types of CFTR protein modulators, there are now a couple of these variants on the market, and depending on the specific kind of defect in the protein, these modulators can be more or to a limited extent only efficacious. So ivacaftor is a compound which is really activating or working on the channel to open it at the apical membrane, so it helps to open the channel once if it's already expressed at the apical membrane. Lumacaftor is a compound which is really helping the trafficking, for instance, of this F5-ODEL variant, which then helps it to traffic towards the plasma membrane, where it then also can be opened up by ivacaftor. And there's now a new compound on the market, elexacaftor, which is also a corrector compound and also helps to get the F5-ODEL mutation to the cell membrane. What we also know from these types of compounds, I think, especially now the new compounds in which you have the second generation treatment, where also this elexacaftor is used. So actually, it's a triple combination of these three molecules. This can be really efficacious on F5-ODEL, and it's a big question which other types of mutations also may, or which people and carrying which types of mutations that can also benefit actually from this triple combination. So it's a big question in the field, especially for these rare variants, whether people can benefit from these types of drugs or not. So in our lab, we try to study this with patient stem cell culture. So this is just a general workflow which we have in the lab. So we have a large clinical center which we associated with in Utrecht. So we have about 500 patients, I think, in our total center. We generate a lot of organoid-based tissues. So these are these 3D culture systems, either from the airways or from the intestine. We generate these. We store these in living biobanks and use these types of cells for preclinical studies where we look at assay development or pathophysiological aspects of disease, or preclinical drug efficacy studies or drug mode of action studies. But we also use them as ex vivo representatives in the lab of individual patients. So you can, so in this way, you can try to see whether you can well, recapitulate clinical features in these types of models and be also very precise in measuring specific features of the disease, like we do here also in my talk, which I'll be showing that we measure CFTR function in these types of airway cultures, and also in the intestinal cultures. So this is relevant for diagnostics, but also for trying to classify disease severity, or potentially also for selecting individual therapeutics. But you can also think that it could be an endpoint in a clinical trial. So just the structure of the talk, I'll be discussing a little bit on what we do with intestinal organoids, how we measure CFTR function, how these types of functional assays relate to clinical parameters, and how once we establish that there's a good relation between in vivo and ex vivo experiments, we are now trying to really use this model to characterize rare or organoids from patients with rare mutations throughout Europe, to see whether we can make sure that these people may also benefit from existing drug development programs. And I'll also be showing or sharing some data which is still unpublished, but on a new mode of action of this elexacaftor, which is currently being in the process of approval by EMA and the various countries in Europe. I'll be also briefly talking about airway organoid studies, but I will just see how far we go with time. So just something general on organoids. I think this was a seminal paper in 2009 by Toshi Sato from the Clavis group, where he showed that you could use a single stem cell. This was all mouse in the beginning, that you can use a single stem cell, and he identified a culture growth medium, or a mix of growth components, which you can just simply add to the cells and the cells would just then grow out into a self-organizing complex tissues, which had different types of differentiated cells, and also various domains like grip structures, which were located here in these buds, but also villus-like domains, which were located in between the buds. And one of the beauties of this system was that the organization from a single cell towards a complex structure in which you see architectural features of the normal tissue in vivo. This is all a self-organizing system. So it was a pretty simple way to get a complex structure in vitro. And another thing which was really powerful, I think, was that these cells keep on growing for a long time. So only by adding growth factors you can really have a long-term primary cell culture in your lab. This was also developed by Toshi for human cultures and somewhat later. And this is where we also started to work with CF and these types of models. So we already knew from work actually of Marcus and Hugo de Jonge in the Netherlands that rectal tissue is very relevant for CF. There's a high CFTR function which can be measured in rectal tissue. So we reasoned we were also getting these rectal biopsies in our lab for CFTR function studies. So we thought that this might be a good way to start these cultures and see whether we could measure CFTR function as well. So in human conditions, we don't use single stem cells, but we actually isolate tissue fragments, which you then put in a 3D culture medium. So you use Matrigel to recapitulate the extracellular matrix, and you use a couple of growth components, arspondin, Win3A, and Noggin, various others, to make sure that these crypts start to close actually, and then start to grow. And the apical membrane is here on the luminal side of the structure. It's a single epithelial culture model. It's only epithelial cells, and it has a tissue architecture which resembles some features of the in vivo tissue. And also for the human system, again, this is really a powerful tool, because just by adding a couple of growth components, you can keep on culturing this. So these are tissues which are relatively genome stable during culture, and you can keep them, you can grow them for easily for over a year or over 2 years. So it's really something which you can propagate and would allow you to culture individual cells and make biobanks of this in your lab. So we rapidly discovered already that healthy control organoids and CF organoids are very different. And this was mostly also dominant when we activate the CFTR protein using a cyclic AMP agonist, forskolin, which raises cyclic AMP, activates protein kinase A, which leads to phosphorylation of CFTR, and it's the opening of the channel actually. So this, and if you incubate organoids, these intestinal organoids with forskolin, you get these fluid transport into the lumen of the organoid. And these are movies of about 60 minutes. What you see here is that these types of organoids, if you activate forskolin, they pump in fluid and they swell up because like little water balloons actually. And one of the beauties of the system was pretty surprising actually. If you do this with CF organoids where you have mutations which completely disrupt the protein function, so premature stop codons, for instance, this swelling is fully absent. Also in the mouse knockout, it was fully absent. So it really showed that this forskolin-induced swelling of these organoids was a CFTR-dependent assay. So what we do here is, so what happens, so you have an organoid, you add forskolin, which activates CFTR, you get fluid transport into the lumen, and this leads to organoid swelling. We quantitate this using live confocal microscopy. That's all published, so I won't go into details there. And we were really started to get exciting because this was in the time that these new CFTR modulators were entering the market in a very narrow disease category still. There were for several mutations, it was clear that these drugs could be effective, but for many rare mutations, it was still completely unknown, and lots of people were not having access to these types of modulators. So we obviously thought that this would be a nice tool to use to see whether people may or may not benefit from these types of new therapeutics. This just shows you how these organoids respond to these types of therapeutics. So this is ivacaftor lumacaftor combination treatment for organoids from a person with two severe F5 mutations. So this is without treatment. You see that the organoids show a little tiny fluid response. So there's a little tiny bit of residual function. It's a very sensitive system. So this is that you can see this is already indicating that it's a sensitive readout, because there's hardly any CFTR function in these types of mutant organoids. But if you then start to incubate with these types of CFTR modulators, you could restore the swelling again. And then simply by measuring swelling, it allows you to measure individual responses to CFTR modulators. So it was a very simple, straightforward readout, which we could do also in 96 well plates instead of the conventional Assing chamber systems, which are very lengthy protocols. So this would have really allowed us to do these types of measurements at a larger scale as compared to the current at the standard of assays which were then which were present at that time. So a big deal of our work then shifted to a really translational question. So I had a molecular background, but we really focused with the physicians also on, well, what we now see in vitro, is this also really corresponding to what's happening in vivo? And just because of the time, I will be a bit quick here, because we published all these types of, we published this all, and I would just like to focus a little bit on some of the new data which is still not published. So we looked at modulator responses in vivo and in vitro. These are just some proof of concept studies where we looked at, where we showed that even over really long, prolonged time of culturing, patient individual responses. So this is all organoids from the same patient cultured over a long time period, also including biobanking. You see that more or less the patient-specific responses are retained. So this was an important observation. We also found that organoids from people with different mutations show also different responses to drugs. So this is a mutation phenotype which is responsive to both lumacaftor and ivacaftor. If you just look at the patterns here, this is a patient sample which is a gating mutation, very much responsive to ivacaftor, but there's hardly any response to lumacaftor. Also, the combination doesn't do much more than ivacaftor alone. And here we see an opposite response as compared to this one here. Or an opposite response here, we see really a nice lumacaftor responsive genotype, so E92K. So this just showed that we could use this in a mutation-specific manner. And we then did a lot of observations between what's happening in vitro and what's happening in vivo. And I'll just briefly, we looked at correlations between two of the most important, I think, clinical or efficacy markers for CFTR modulators in clinical studies. So this is FEV1, which you use as a surrogate to measure pulmonary function. And we also looked at sweat chloride concentration. So this is the best established in vivo biomarker of CFTR function. And both these markers are very helpful, I think, in classical group-based studies. But there's a pretty large impact also of non-CFTR dependent factors, which can modify or modulate the reading in these types of endpoints. So we compared this with phosphoryl-induced swelling of these organoids, and phosphoryl-induced swelling is nice because it's really a fully CFTR-dependent readout, but it's obviously an in vitro model, so you don't have any PK information. If you have differences between PK between patients, you don't incorporate this, obviously, and you may also modify the tissue by culturing it in vitro. But we were really happy to see actually that these cultures, which so that swelling in organoids, if you just look at it, this is a sample set of about 34 treatments, if I remember correctly. I don't know, 34, 35, something in that similar size, and these were derived from 24 patient samples. And in general, you see the better swelling you have in vitro, the better a response you also see in the pulmonary domain. We see similar relations also with sweat chloride concentration. So if you have a large readout, you have a large effect size in your organoids and also a large effect size in your clinical parameters, it's nice, and you can see these types of correlations between organoids and clinical parameters. I think one of the challenges, if you think about doing these types of clinical studies, what this is also known in the field, and it's perhaps well, it's not so easy to correlate actually individual clinical responses. So if you just look at the clinical parameters in these types of cohorts, you see that people who respond in FEV1 and the correlation to the response also in sweat chloride there's a trend, but it's not so strong actually. So this indicates that there's significant noise or variability within these clinical parameters, which makes the individual measurement, the accurate individual measurement of treatment response pretty difficult actually. And I think it's also shown here. If you just look at the red bars here, this was the first study by Boyle et al. Looking at Lumac after treatment, and this is the variability in the. So this is the placebo treatment group, and this is the variability between individuals. So you see that people, even in the placebo group, there's a lot of variability in FEV1, and obviously this makes it more difficult to make really tight correlations. So I think there's still a lot to do if you think about clinical or measuring accurate of measuring the clinical response to a modulator at the individual level with high accuracy. I think that's still pretty challenging. But we've done a couple of studies now. We looked at residual function. I think also at modulator responses, also together with Marcus Mall, Simon Graeber. I think in general, if you look at these types of studies, the bigger effect sizes you can measure across your correlation data set. If you have big effect sizes, then you can nicely make correlations. If the effect sizes are smaller, like this study here, in which we look at effect sizes of a treatment which is not really highly effective, so this allows you to have a lot of noise also, I think, in your clinical parameters, and also the technical or some noise in the organoid measurements as well, because of technical technical variability. You see, we didn't observe a clinical or a correlation here between the individual data, although if you look at these larger effect sizes, it nicely correlates. If we move on from this, all these studies up until now, which we have published, were associations between a short-term disease development, so for newborns, for instance, or for short-term drug responses. So obviously, what we also would really like to know whether these types of organoids can also predict or have a correlation with long-term clinical phenotypes. So we've done this now we haven't done this for drugs yet, because we don't have the drugs for a long time available already. We've done this a little bit, but not on a large scale. So we looked actually now at the first long-term clinical data sets is the relation between residual function, so without any treatment, and clinical phenotype. So what we do here is we collected, I think we have collected now more or less about 40% of patients, CF patients, or 40% of Dutch CF patients, we have collected the organoids from them. We don't have informed consent from all of them to link to Dutch registries, but we had it from 170 subjects more or less. And we looked at, for these 170 subjects, we looked at the annual decline in ppFEV1, for instance, but also comorbidities like liver disease, pancreas function, and diabetes. This is still all unpublished. I'm really not a statistician. We used so this I always have to rely on the people who do the math and the statistics. Danya is really good in these types of things, but we used a linear mixed effect model with random intercept and random slope, and several fixed effects like organoids. But the goal here obviously was to look at various potential predictors like organoids or sweat chloride levels or genotype or sex, and how this relates then to these various clinical phenotypes. This is how the organoid responses look like in the cohort. All the color schemes are different types of mutations. But I think the most important message here is that residual function across all these types of organoids is pretty continuous scale from very little, so no residual function. This is where you see stop codon mutations, F5 del homozygous mutations, and so on, towards pretty high levels of residual function actually, which correlate with splice mutations, not in the consensus sites, but far away from the intron exon boundaries or specific types of missense mutations. So if we look at results or correlation between organoid function, so residual function in the absence of treatment and FEV1 decline, I think this was really well, exciting data. If we look at these, so age obviously is something which is relevant for decline in FEV1, but also if we correct for age, the organoid measurements are also very strongly connected to FEV1 decline per year. So what we measure is if in organoids of people without any phospholipid-induced swelling, the average FEV1 decline per year, the annual is minus 1.2, but for every 1,000 units of FIS, this drops by about 0.35%. So this was a really nice linear relation across the data set. So this really showed now that also annual decline per year in pulmonary function is related to organoid swelling. So this was nice because this was the first time that we really look at long-term clinical disease. More or less, so I'll just do this quickly. This you would expect this, at least I would expect it. Pancreatic insufficiency is also a clinical phenotype of CF, very much related to CFTR, to organoid swelling and CFTR residual function. So high chance for pancreatic insufficiency if you hardly have any organoid function, while it drops down if you go to higher levels of function. Same is true for CFTR-related liver disease. The probability scores are a bit lower, but again, a clear relation between organoid function and this comorbidity. And the same also holds true for CF-related diabetes. And within this phenotype, there's also a component of age. But you see for the various different age groups, that especially if you are older, you get a high probability of developing CF-related diabetes if you have hardly any FIS, or if you have these higher amounts of residual function or organoid measurements, there's hardly any chance of developing CF-related diabetes. So this was nice. It shows that we have an annual long-term FEV1 decline association with these organoid readouts. We also looked for association of this with the sweat chloride concentration in the cohort, but there we didn't find an association. It was a limited data set of only about 100 subjects because we didn't have sweat chloride concentration from all subjects actually. And it shows, I think this is perhaps a take-home measurements, that for residual function measurements in these organoids per 1,000 units of swelling, we see the annual PP FEV1 decline lowering by about 0.35%. So this is really nice. It doesn't translate. I don't think that you can translate this directly to drug-induced function. So we just have to learn and see how 1,000 units in area or swell increase by drug-induced function, how this will correlate with long-term benefit. This is just something we still need to do at a larger scale. So just to quickly sum up some of these conclusions here, in vitro organoid FIS correlates with these in vivo disease or drug response indicators. For modulators, I think especially if you look at in vivo measurements, the accurate definition of clinical responders remains still challenging, but especially if you have these larger effect sizes, then there's a really nice correlation between what happens in vivo and also what happens in vitro. There's some caveats here. So the drugs we used were still, relatively small scale studies, biased towards potentiators and so on. And well, so also indicated the organoid measurements is pretty sensitive. So you have a dynamic range between CF to borderline CF, but it's very difficult to correlate or to look at responses as a percentage of wild type, for instance. So just to go quickly over a couple of interesting ideas here. So we just showed that or what you measure in the dish in vitro is corresponding with what happens also in vivo. So we reasoned a couple of years ago that there's a large unmet need for people with these really rare mutations, and we got a European grant also to look at these rare mutations and see whether we could also get access for people with rare mutations to these new types of CFTR modulators. So this grant was all about trying to generate therapeutic options for people with these rare variants, use intestinal organoids as a qualified diagnostic for drug access, and also then if we generate these organoids from all these people with rare variants which don't have access to current therapeutics, can we use them and store them also in a biobank? So this process is still ongoing. There are two stages. So one is the first stage is to collect 500 biopsies from patients with rare mutations, which are not eligible for any other CFTR modulator treatment. Then use these modulators in a preclinical screening setting to test drugs of various pharmaceutical companies, and then select the high responders and then do clinical trials. So mostly this is the part where we're now at, and we are trying to see whether we can start and move on to this part at the moment. This is just a distribution of, if you look at ECFS patient registry data, this is the distribution of people with two rare CFTR variants across Europe. So obviously these are people with rare variants, but as a whole, the total eligible patients in Europe is about 2,000 adults with really rare mutations which do not have any access to the current CFTR modulators which are on the market or still in development. And it was really nice that we could, well, I think this is really nice, success that from across the majority of countries we were able to recruit patient samples. So this is really a long or a huge effort by the CF community to ship biopsies actually towards Utrecht, where they then were processed for organoids and for testing within various laboratories. And I think this is a really also pretty interesting observation. So if you think about doing these types of studies at a large European scale, also I'm amazed by the robustness of these types of cultures. So we have 500 biopsies shipping in from all across Europe towards Utrecht. They were processed by Hub for organoid generation, and about 95% of samples can be successfully turned into an organoid. So even with shipping from all these various different sites, with all these different people taking biopsies, it's still a very robust model. So it's really something which you can do at a large scale and be successful in generating organoids, which then are useful for these types of measurements. This just shows some of the early screening efforts. So here we see about 230 organoids which are responding to a specific drug this is actually the desivacaftor, but this is these are drugs in development by Proteostasis Therapeutics, and they have this. Here we just did the screening. There's some technical reasons why you have negative values here. That's not, but you see that there's a good proportion of patients with rare mutations showing an equal or higher response as compared to Tez-Iva, which is a current standard of treatment in people with F5-odel homozygous mutation. So there's a good proportion of people responding to these types of treatments. And also, if you look at the positive ones, actually this is also pretty nice. So we looked here at responses. So for all these mutations, for all these patient samples, we also looked at the Tez-Iva response. So this is the current treatment which is available for people with F5-odel homozygous. So if you look at the distribution of people who are either responsive to Tez-Iva or to the potentiator corrector combination of proteostasis or both, you see that there's a good set of mutations or genotypes or patient samples which is responsive both to the combination treatment of PTI and also the treatment of Vertex. There are some mutations which are preferentially responsive to the Vertex treatment, whereas other mutations or patient samples are preferentially responsive to the PTI combo. So this I think illustrates that these drugs have a different mode of action, and that there also are people who can benefit potentially more from this combination as compared to a current drug which is already on the market. So I think it indicates that it will be helpful if we think about optimal treatment for all these people with CF, that we need more products on the market so we have more modes of action available to treat the wide variety of mutations which are out there. So just to quickly summarize this part, so it's feasible to organize a centralized effort to collect and measure organoid responses from people with rare disease. You can standardize this across sites. I haven't shown this, but we do this together with Leuven and Lissabon, Margarita Amaral and Chris de Boek Lab. It shows a nice reproducibility, and we're almost finalizing the screening efforts now, and we see these differential responses between different types of modulator combinations. Just because of the time, I will just go quickly here. I think we try to publish this now. We look at this. There's this new modulator on the market, which is still not here available in Europe, but it's out there in the US. I think I'll just skip. So this just confirms that this VX445, which we call this modulator, is a strong corrector, which we know already also from the published literature. What we also find, and this is, I think, for the CF people among us, is pretty interesting. We see also if we use this as an acute stimulus, which indicates that it has potentiator activity, we also see a very strong effect on specific types of mutations. So this just shows that apart from being a corrector, VX445 is also a potentiator. We also see, if you look at combinations of two potentiators, so 445 using in combination with the known potentiator VX770, that the combination is very strongly synergistic as well for certain mutations. So it shows that even for people who only have gating mutations and no corrector or no trafficking mutation, that people who may not benefit fully from ivacaftor, which is already on the market for gating mutations, they don't fully benefit. You may also can think about using Trikafta as a treatment to really boost the potentiator activity of treatment. So if we, I'll just perhaps, yeah, I think if we just summarize the part, we work on airway culture models as well. I think what we really, what our current standard is to still, we rely pretty heavily still on ALI cultures, which are more or less conventional cultures, but we have nice protocols to also generate airway organoids from these ALI cultures. And we do this because you get a more homogeneous response and assay model as well. If you generate airway organoids directly from tissue, you see a lot of different structures which have a different differentiation status. So there are organoids which are mostly basal cells, there are other ones which are highly differentiated. If you do it like this, you really get a nice homogeneous organoid. So these airway organoids, you see they also have the ciliated cells here on the inside. You can nicely also well image them with confocal microscopy. But we are really struggling to really get a nice functional assay also with these airway organoids. And now I think we now have nice protocols where we can look at, these are nasal cells from people with F5ODEL, homozygous CFTR mutation. So you see it's pretty homogeneous. So you see all these types of organoids which have, most of them have a nice lumen, hardly any response in the absence of treatment. But then if we do these Trikafta treatments, you really see nicely that they also start to swell. So we like this because obviously the CFTR function and the interaction with other ion channels can be different between the intestine and the airways, and this also allows you to just use a simple nasal brushing to look at patient individual modulator responses. So I think with this being said, I can keep on going, obviously, but I think this is also nice. If you think so, what makes an organoid really helpful? If you think about a conventional ALI culture, for ALI cultures, it takes you obviously a couple of weeks to differentiate. But from only 4 filters, you can fill a full 384 well plate of organoids, and then you can use these 384 well plate settings for larger scale drug development screening. So we use a deep learning, we modify the deep learning algorithm to really nicely pinpoint all these types of organoids in our dishes, and then you can do screening, and this is just to screen for fluid secreting agonists. But you can also really nicely see that in these types of settings, you have a very nice baseline here and a good signal to noise ratio if you think about hits which are identified here. So just to illustrate that these types of screening models can be used also at a larger scale, and this is all primary cells which you can now also grow at a large scale and do these types of screens. I think even with it would be feasible to do 100,000 compounds or so, or even more. But just to summarize here, for airways, it's growing biobank. I understand I went quickly here. I'm really pretty glad now that we get good protocols also for nasal cells. We have, obviously, nasal cells allow you to have easy access to tissue, but it's still a little bit limited in expansion if you would compare it to intestinal organoids, and also the robustness is not as high. We are now at a larger scale doing these in vitro and vivo measurements also for nasal cells, and it works also in 384 well plate setting. So just in general, I think if you look at the value of primary cell models, these new types of cell models which you can generate with high efficacy from individuals, it really gives you a way to pinpoint at high precision what a specific biological process. So it really is a nice opportunity to do research in a clinical context. So it's feasible to set up large-scale screening infrastructures. You can process large patient numbers. There's still some work to be done to get these types of models accepted by regulators and payers. And obviously there are also ethics and involved if you start to use these types of models from individuals. I think especially for monogenic diseases, these types of cell systems are really relevant. And using a variety of assay protocols, you can really accommodate a variety of research questions. So with this being said, lots of people to thank, but I'm also a little bit late. So perhaps there's some time for additional questions. And I thank you for your attention.
Thank you, Jeff, for a great and talk and tour de force really through your research, and that's obviously developed very nicely, and you really spearheaded this topic in the CF world and community. I don't see any questions yet through the chat, but we still have some minutes, so if you have questions, please send them through, and I will probably get started. So with your, as you mentioned, the correlation between your forskolin-induced swelling and the registry data. Yeah, I think that's really, fascinating in terms of predicting long-term, as you say, long-term outcome. What was not so clear to me was this registry data from across all genotypes. Yeah, and so that's sort of what I suspected. And so if you look at the delta F508 homozygous patients only, can you then also start to see a correlation, or is the sample size too small so far?
Yeah, no, it's a very good one, but it's mostly a data set which was enriched for more rare mutations. So it's something which we should be able to do. I think we have a large cohort of F508 homozygous patients where we looked at Orkambi responses. I actually like the study which we published with you, Marcus, also, but at 450 patients. There we haven't also we don't see a clear correlation, and we haven't done this for long-term disease. It's something which we should be looking at, and I think it's probably there, but whether it's useful for individual clinical purposes, I don't think. I think there will be a correlation, but there within these types of differences of CFTR function, it's still pretty small. And I think the impact of immune modifiers in the immune system and just environmental challenges, that will be bigger than the CFTR variability. I think that would be my prediction, but it probably will be there to some extent.
Yeah. Well, let me follow up on this regarding variability. I mean, you obviously you mentioned that it's difficult to, and one issue in correlating the functional responses in the organoids to clinical data is the high variability in the clinical data. Yeah, especially if we look at lung function, that obviously very much depends on the stage and severity of the disease. But on the other side, there's also, as you mentioned, some variability in the CFTR function, even in patients with the same CFTR genotype, even in delta F5 homozygotes. So do you think there's also an opportunity to use your organoid system to try to get at potential disease modifiers?
Yeah, I think so. So we have a couple of lines of research here. If you think about disease, especially in intestinal organoids, we measure it's very CFTR dependent. So if you have variability there, I didn't include it in the presentation because it's just it was already pretty dense. But we have a very nice protocol now to haplotype the full CFTR gene. So we can just, without doing analysis on parent samples, we can just now have a sequence protocol, pretty complex, which allow you to look at haplotype variability. So one of the sources of variability may potentially be within the CFTR gene itself. This is something which you may be able to look and quantitate in the intestinal organoid. Another variability what could work would be other ion channels. It's difficult to look at in these intestinal organoids because it's so CFTR dependent. But this is where airway organoids, for instance, come in handy, because depending on how you differentiate the cells, there's more variability. If you generate lots of mucus-producing cells versus high levels of ciliated cells, you see other components, other ion channels being expressed, and there you can also look at functional responses in the airway cells. And it's something actually, Marcus, which we should do, because I'm also the airway cells are even the primary cells are very easy to generate gene knockouts actually. So it's just a simple transfection which allows you to knock out about 90–95% of the cells for a specific gene. So we're now having TMEM16A knockouts. We have SLC26A9 knockouts. We have a couple of other ion channel knockouts as well, just to look at whether we can develop assays in these primary airway cells for the various types of ion channels, which can also contribute to a modifier or to differences in fluid secretion.
Yeah. Jeff, we have an interesting question here, and I'm not sure if you have looked on it, and that is, how do mechanical cues such as pressure or density of the extracellular matrix impact on the function of intestinal or lung organoids in the context of CF?
Yeah, it's a very good question, and I don't know. I think if you look at these long on a chip type of systems where you have a flexible membrane on which you put the epithelium, you can play around with these types of pressures, but we haven't done this so far. What we do sometimes is dilute the matrigel to various different extents. There you don't see so much of a difference. So if you make a 50% Matrigel solution or a 25% Matrigel solution, you see them more or less being similar responsive, albeit that we haven't looked in so much detail. If we do these types of dilutions, it's mostly also because of monetary reasons. If we do really these large scale screens, it helps if you can dilute the Matrigel a little bit more, for instance. But it's something which is interesting. But I think these long on a chip models could be really helpful there, because then you can perhaps more precisely manipulate the tension across the epithelium.
Okay, thank you. I have one last question, because that's more so in the clinical direction. So regarding the HIT-CF study, when you showed the map, there were many more, or there were less rare mutations in the southern part of Europe than the northern part, and this is sort of the inverse of what we think. But this is probably related to less extensive genotyping, I suspect, in these countries.
Yeah.
Then the second question, I think there were plans that the HIT-CF study should already be probably up and running at this time, and it has been delayed due to the pandemic. But do you already know if and when and with which compounds it can get started?
Yeah, it's also a good point. So I think if you look at the distribution of rare mutations, Italy pops out. So Italy has a lot of patients with rare mutations. I think what you indicate is correct, right? So I think a lot of perhaps the registries are not fully genotyped or complete. So I think this could also be an important factor, that it's not only that it's a bit limited perhaps in several southern European countries. With regard to there are a couple of, there are definitely issues with COVID now. So this delays a lot of things.
Yeah, I think that's maybe also an important, almost concluding remark, that in the pandemic, this is not only challenging for providing clinical care for patients with cystic fibrosis and other chronic conditions in a lockdown and limited access to hospitals, but that it also has an impact, a huge impact on clinical trials. And if we know how long clinical trials take, mean a delay of several years of new drugs, or as you mentioned, potentially the discontinuation of a promising program. So thank you again. And I would also like to thank the audience